This is the fourth chapter in system of general physiology, membrane potential. Now, when we see this term as membrane potential, the potential that created at cell membrane level. In first few lectures, we see the basic structure of cell membrane, the basic functioning or basic functions of cell membrane. At that cell membrane, different transport processes respect to the active transport and passive transports. Toward the end, when we see the resting membrane potential that is observed in all cells, while the action potential that is nothing but impulse that is observed in excitable cell tissues. The resting membrane potential, it is nothing but it is a negative on inner side as compared to outer side of the cell membrane. That means resting membrane potential is a polarized. Now next question arises in our mind, that is how this resting membrane potential occurs, or what is the genesis of resting membrane potential. First, properties of membrane that related to the resting membrane potential, respectively potassium permeability, Donan's effect, sodium potassium pump, and some permeability for other ions. Secondly, the role of potassium concentration gradient. Third is presence of leaky sodium channels, and fourth, the role of sodium potassium pump. These are the basically four factors which are responsible for the genesis of resting membrane potential. Now the second concept, that is action potential. The action potentials are the changes in membrane potentials produced by the application of adequate stimulus to an excitable tissue. As a result of the application of adequate stimulus which creates changes across membrane level and which creates us impulse. This impulse is nothing but action potential. It is very clear and easier concept. Once you understand this changes or this impulse which is known as action potential, these changes occur during action potential, that is depolarization followed by repolarization of the membrane. This is the basic change that is observed, that excitably observed as a depolarization which is followed by repolarization of that membrane. This is a simple diagram that explains you that process, how it goes, depolarization followed by repolarization. When we see the different parts of this graph of action potential, these are divided into different phases. First important phase is stimulus artifact. That is just immediately application of the stimulus, there is observed some excitation or some changes. These are stimulus artifact. Subsequently, second is latent period. Following that stimulation, some phase goes empty, which is known as latent period. Third, local potential, followed by days of the rapid depolarization, which is followed by rapid repolarization. Now the phase that is followed by rapid depolarization, followed by rapid repolarization, which is known as overshoot, and follow where there is observed after depolarization, following by after hyperpolarization. Toward the end, what are the methods of measurement of resting membrane potential, which we'll see in next slide or next lecture series.